All right, this is Microbiology Lecture 14, Part 2. We're continuing with principles of infectious disease and transmission, epidemiology. Now, transmission is, of course, an important aspect of any infectious disease. It's important to understand how the disease gets transmitted. After all, infectious disease cannot, from the point of view of the organism, the microorganism causing the disease, it cannot succeed unless it gets transmitted from one susceptible individual to another. It doesn't have to do that every single time it infects someone, but it has to do it overall, and it has to do it consistently, otherwise it would disappear from the population. So there are different kinds of contact transmission. There's direct contact transmission that requires close association between the infected and susceptible host. That's person to person. There's many organisms that do that. There is indirect contact transmission where it is spread by non-living objects. And these non-living objects are classified, they're called fomites. So this would be, for example, something that's transmitted by needles that get infected, or with a virus, for example, like hepatitis B. Very common way that hepatitis B gets transmitted from individual to individual through a fomite, a needle. There's also droplet transmission, which is doesn't involve any non-living object, but is technically a subcategory of direct transmission, but not through contact. Direct contact. It's droplets that are released during coughing, spitting, talking, yelling, chanting, singing, praying, anything where you make increased, I should have left out praying, anything where you make any sound at all, extra effort coming out of your mouth, you're always releasing aerosols. And that can be seen here. Here's a direct type of contact transmission. This is potentially transmission by droplets. You release a huge amount of droplets. This is a sneeze. Quite terrifying, really, how much gets released just by talking. There are other kinds of transmissions. Some involve what's called vehicle transmission, where it's spread by an inanimate reservoir. There is a reservoir that holds the organism, and it's inanimate, not a living thing or another person, and it spreads it. And this includes food, water, or air. Vector transmissions are mediated typically by insects or arthropods, and some examples of insects that can do that. Not all the ones that are out there, but fleas, ticks, mosquitoes, they're all types of organisms, and it's usually a certain species of these for each of the different diseases. But fleas, ticks, mosquitoes can all act as insect vectors for transmission. Now, an insect vector could transmit mechanically, so you can have mechanical transmission mediated by a vector, and that's when the insect carries the organism on its feet and touches the food. It goes from something that's contaminated to the food, and then contaminates that, and then you eat the food. That's a problem. Or it could involve a biological transmission. Now, in biological transmission, the infectious organism, the pathogen, is reproducing in the vector. And an example of that would be malaria, where the protozoum Plasmodium vivax, for example, which can cause malaria, is reproducing in the salivary glands of the mosquito. And here you see a type of mechanical transmission. I was once doing tissue culture with different types of cells, human cells, and in a containment hood where everything is sterile and the flow of air is very carefully controlled, but it has an open front, but any air that tries to get in gets sucked down because there's a grid right at the front of the hood where you're working that pulls everything in. And a fly flew in and landed right on what I was working. It was just unbelievable. Here is not necessarily an example of transmission, but it could be. It could be the surface that here, the surface here is not perhaps clean properly, or the person's hands are not clean, or something for some other, or the person coughs on it and aerosolizes whatever it is onto the food. There's all kinds of risks involved. All right, let's talk about something closer to home and closer to particularly relevant to you, which are nosocomial infections, which are extremely common and really a large problem. These are hospital-acquired infections, all of which could be preventable. And really, a big part of this course is making you aware of how the infectivity and the ability to transmit disease makes it imperative that when you're working in a hospital environment with sick people, immunosuppressed people, people who have infections, that you don't spread it around, that you're not making things worse as opposed to helping their health improve. Nosocomial infections are infections acquired within a hospital as a result of the stay in the hospital person. Hospital long-term care facilities are major reservoirs of infectious disease. Many people in them have infectious disease, and any stay in these kind of facilities, either hospitals or in Quebec it's CHSLDs, carries an increased risk for the person catching an infection. Most microorganisms that cause nosocomial infections, this is an important point, are harmless to a normal person. So you guys go in and you may pick it up, but you don't transmit it. Yeah, that's not right. You can transmit it, but you don't come down with any disease from most of these microorganisms that are picked up in the hospital. In fact, when you make a survey of people who work regularly in the hospitals, then a high percentage of them will show, in fact, MRSA, for example, in their nose because they pick it up from patients, and that's their environment. That's part of the environment that they spend time in, so it's possible to pick it up. So for a normal person, healthy person, most nosocomial infections or nosocomial organisms are harmless, but that does not mean it can't get passed on. About 5 to 15% of all patients in care facilities or hospitals, in fact, do pick up nosocomial infections. It's a big problem and increasing in numbers. There's more than 200,000 cases per year in Canada, and more than 8,000 deaths occur due to that. It's a constantly changing, evolving situation. Hospitals typically have an infectious disease control team to educate and monitor and try and prevent nosocomial infections. And different facilities or different institutions have different percentages of cases. Nosocomial infections occur as a result of three overlapping factors. One is the microorganisms that are present within the hospital environment. We've spoken several times about this. Another is the presence of a compromised host. And it's the compromised hosts that will pick up the infection and get an active infection. Will pick up the organism and get an active infection. And then there has to be some sort of chain of transmission that spreads it throughout the institution. And so where these three things overlap, that's where you get cases occurring. And the more they overlap, the more the cases are. The more microorganisms in the hospital, the more immunocompromised individuals there are, the worse the chain of transmission is. It can take something as simple as someone who just does not understand the idea of using proper aseptic technique to cause a spread of infections widely in an institute or in the area where they work. Just look at people who refuse to wear masks for a respiratory infection. Okay, so in terms of microorganisms in the hospital, as I said, the hospital has it's an environment, it has its own ecology, its own set of organisms that you find there. There are many antibiotic resistant microbes in the hospital environment, many more than elsewhere because there's a lot more antibiotics used. So the odds of finding antibiotic resistant ones are much higher. These are opportunistic microbes that will cause disease in immunosuppressed individuals. But overall, these are all part of the normal microbiota in the hospital staff and personnel, and in many patients who don't get the infection, but again, they just carry it. They'll pick it up and carry it. In terms of compromised hosts, it's not just being immunosuppressed, but anything that lowers resistance to infection will compromise the individual and make it possible that they may get infected. In this case, normally completely harmless microbes, harmless to a normal person, become pathogens. The main things that are factors that could compromise a host are a break in the external surface barrier, and many patients do have that. And their skin or mucous membranes because of burns or surgery or trauma or injection or IVs, catheters, etc., etc. All of these provide an entry point. The other thing, of course, is suppression of the immune system, immunosuppression, and that can occur with all of these things. I'm not going to just read it out to you, and you can look at it. All of those, all these factors can result in immunosuppression, and when you combine individual with immunosuppression, and many of them have breaks in their external barrier, their epithelial barriers, and you put them into an environment where there's lots of microorganisms that are potentially opportunistic, then you have a problem. If you include a chain of transmission, then you'll start getting serious nosocomial infections. All institutions have them. They vary, but there's significant amounts. Main route is by direct contact, hospital staff to patient or patient to patient. Fomites are a big part of transmission. For example, urinary catheters are a common source for urinary tract infections. When we look at nosocomial infections, the kind of organisms that cause them often are gram-positive organisms. The next most common are gram-negatives. C. difficile, this percentage varies depending on the institute. Probably in many institutes now it's lower, but it's still a problem. Still a significant amount of percentage of the nosocomial infections that are occurring. and fungi is, but all these percentages are, this is the breakdown of the 5 to 15% of nosocomial infections that occur. About a third are gram positives, another third, slightly less maybe, are gram negatives, and then half of that is, well, that would be about 1/6 Clostridium difficile, and then 10% fungi. Many of the gram positives and a significant number of gram negative organisms, bacterial organisms that cause nosocomial infections, also have resistance to antibiotics. When you look at the main site of infection, nosocomial infections, number one by far, urinary tract infections. Patients pick these up very commonly. Surgical site. Lower respiratory infections, that means from the epiglottis down is the lower respiratory tract. So lower respiratory infections here, 13%. Let's get another color, 13% here. But by far number one is urinary tract infections and surgical site of sites. Bacteremia caused by IV catheters is not insignificant, and then other sources or other sites. How are nosocomial infections prevented? By using proper aseptic technique. Whenever you're dealing with anything that could potentially come into contact for a longer term with the patient's mucosal membranes or external tissue, or anything that would pierce through that, like a needle, using proper aseptic technique is vital. Proper handling of contaminated materials, not touching them to anything else and making the contamination wider. Proper hand washing, and just simple awareness is a critical or crucial importance. If you are aware and stay aware, and it doesn't mean you have to be obsessive about it, you just make certain precautions are always routine and you always follow them. So you don't touch something with your hand that you're going to touch onto a patient and then inject. You don't touch the end. You don't touch the needle to something else. You don't. Even when you disinfect a site of injection with alcohol, you don't feel with your bare finger for the vein after that. Feel for the vein and then wipe it clean. The last thing you do before you stick a needle in is you wipe it clean. Feel all you want with your finger, but wipe it with the alcohol and then put it in, not the other way around, which I've seen many times. The other control mechanism that that's in place in all hospitals, there's monitoring of the environment, and that mostly is done by the infection control staff. They keep the statistics, but the environment is monitored, and the patients are monitored, and even the staff is monitored. They take nasal swabs regularly. Okay, as you know, there are emerging infectious diseases, and I've left one out that's important. New diseases or newly occurring in human diseases or suddenly increasing. These are new kinds of diseases new kinds of diseases would include SARS-CoV-2, and SARS back in 2003 certainly was also a completely new disease in humans. Diseases that are increasing in incidence Well, AIDS is still present. I don't know how much it's increasing. West Nile virus, certainly infections with VRE, vancomycin-resistant Enterococcus, or methicillin-resistant Staphylococcus aureus, MRSA, C. difficile, still a problem. Disease that have a potential to increase are TB, cases of TB in AIDS patients. That's a problem. So emerging infectious disease, there's a number of different reasons why a pathogen can be an emerging infectious disease. It's newly recognized. It was present. It was causing disease, but people didn't know that it was caused by a microorganism. An example of that 25 years ago was the recognition that peptic ulcers and even stomach cancer, epithelial carcinoma in the stomach was can be caused by Helicobacter pylori. So new organisms, new types of infectious diseases that are increasing in incidence in humans, or diseases that show the potential to increase, or a number of different reasons. The things that are contributing factors to this, to the occurrence and emergence of emerging infectious disease, include not all of these for everything, but these can potentially cause an increase in incidence. Inappropriate use of antibiotics, leading to antibiotic-resistant strains of an organism. This would be new types of the organism. Inappropriate use of pesticides, resulting in pesticide-resistant vectors. That's certainly a big problem. Changes in weather pattern, climate. Transportation systems are extremely important contributing factors. West Nile virus was brought to North America probably in a ship, and probably by an infected bird in the cargo hold of a ship, and then spread from New York. Certainly was started in New York and spread from there. SARS spread rapidly, very, very rapidly, to many, I think 26 different countries from its site. SARS-CoV-2 spread worldwide, and transportation, flying, airplane, jet transport is a big factor. Sometimes construction, increasing residential construction in areas can have an effect. Coccidioidomycosis is a fungal disease. It is systemic mycosis, and it is caused by inhalation of the spores. And there's one area in California where the spores are naturally occurring in the earth, and if you have construction going around and a lot of dust, it's a problem. In the 60s, when they were constructing the islands in the middle of the St. Lawrence River for a world exhibition, Expo 67, there was dust being spread all around the city from construction trucks carrying fill, and there were a lot of cases of histoplasmosis because of that. Animal control measures can, if they fail, animal control measures, increasing incidence of deer has caused an increase in Lyme disease. And then there's simply, sadly and totally avoidably, public health failure. And that includes all of these things. And I would actually might add to that SARS-CoV-2 in part as a public health failure. Certainly the ongoing spread of it after it was initially occurred, but the initial occurrence could also have been partly a public health failure if it did originate in markets. It's hard to know where it originated, but HIV, many cases, early cases of HIV were also a public health failure, and hepatitis C in terms of spread through the blood supply, because they knew They knew about it, and they knew ways to prevent it through the blood transfusions, supply of blood transfusion, donor blood, but they failed to carry it out. There were many lawsuits about that. Okay, these are old. I tried. Well, these are old slides. I have to admit, 2002, but they get the point across. Epidemiology is the study of where and when disease occurs and how it occurs, what the cause of spread of infectious disease. It's not just study for infectious disease, but here we're talking about infectious disease. So in terms of Lyme disease, it has been increasing. You can see that it's particularly increasing at this time of year. In the fall, particularly, it starts to rise. There's more transmission in the fall. I'm not going to get into that too much. I'm not going to get into these graphs too much. Let's move on. Epidemiology actually has been around for a long time, the study of how, when, and where disease is occurring. In fact, in London, an outbreak of cholera was mapped, and it was clear that it was in certain areas, and it was particularly being spread in those areas. As I already told you, Ignaz Semmelweis in around 1947 demonstrated through careful observation about how puerperal fever, by the way, is an infection in obstetric cases, women who have just given birth, and caused a very high incidence of mortality in new mothers, which was really a tragedy. And he showed that simple hand washing of the attending staff could prevent most of the incidence of this. Florence Nightingale also did epidemiological studies on the incidence of epidemic typhus and showed that sanitation decreased the incidence. She has a major contribution, her epidemiological insight. So there are many different things, it can be descriptive, analytical, experimental, reporting cases. There are quite a few nationally notifiable diseases. Each case has to be reported. It's not reporting the individual, it's reporting the occurrence of a case of a disease. It's vitally important that this kind of data be collected. Nationally notifiable diseases, reporting of unusual disease occurrences also, you can stop or prevent or start an investigation much quicker if there is a central organization that collects data and can spot increased number of cases of infectious disease. This is just a short listing of many of the notifiable diseases that occur that are notified, so that statistics are kept of the number of cases appearing, and it's on a weekly basis. In the United States, there's the Center for Disease Control. In Canada, and I'll talk about that in a later lecture. But in the United States, the Center for Disease Control publishes weekly morbidity and mortality reports. These are called the MMWR. This is a live link that you can look up and look up MMWR to see what the weekly statistics are for all of the different notifiable diseases. Morbidity refers to incidence and mortality to death. So the morbidity rate is the number of people affected per total population in a given time, and the mortality, the number of deaths per population in a given time. Mortality rate, at least. These are rates. These are just whole numbers.